welcome to our APS classes. This is our biology class and today we will continue with our part number 2 for the chapter number 13, and the name of the chapter is photosynthesis in higher plants. And in our part number 1 we have discussed about the process of the photosynthesis. What do you mean by photosynthesis? तो photosynthesis का process क्या होता है? The synthesis of organic food from carbon dioxide and water in the presence of sunlight is known as photosynthesis. So green plants, they have the capability to make their own food in the presence of sunlight by the process of photosynthesis. So synthesis is the most synthetic process on the earth, because in this process the stored energy, that is carbohydrate, that is transformed the solar energy into the chemical energy, and in other words we can say that light, chlorophyll and water, these are very essential for the process of the photosynthesis, and these can be demonstrated with the help of two experiments, that are variegated leaf experiment as well as light strain experiment. With the help of these two experiments we can demonstrate the process of photosynthesis, that in the process of photosynthesis light, carbon dioxide and chlorophyll is very necessary. And where does the process of the photosynthesis comes from? How many photosynthetic pigments are present in the process of photosynthesis? That means there are mainly three photosynthetic pigments that are present in the plants. That are the chlorophylls, carotenoids and the phycobians. So in part number 1 we have discussed up to the chlorophylls. तो हमने part number 1 में जो है वो up to chlorophylls तक discuss कर लिया था, जिसमें various types के chlorophyll जो थे जो कि photosynthesis के process में present थे, such as chlorophyll A, B, C, D and E, bacteriochlorophylls. All these chlorophylls, they are present in the chlorophylls. And today we will continue with our part number 2 with the topic that is the carotenoids. What do you mean by the process of carotenoids? So in the process of carotenoids, protect chlorophyll from photooxidation. तो जो कैरोटीनॉइड्स की प्रोसेस है इसमें जो है ये क्लोरोफिल को प्रोटेक्ट करते हैं फ्रॉम द फोटो ऑक्सीडेशन से ये क्लोरोफिल को प्रोटेक्ट करते हैं. सो नोन एज द शील्ड पिगमेंट. इस वजह से इनको क्या कहा जाता है? शील्ड पिगमेंट कहा जाता है. दे आर येलो ऑरेंज पिगमेंट्स. अब कैरोटीनॉइड जो है इनका जो कलर है दैट मींस दे आर येलो ऑरेंज पिगमेंट्स, वाटर इनसॉल्युबल एसेसरी पिगमेंट्स, और ये वाटर इनसॉल्युबल एसेसरी पिगमेंट्स हैं. No light is required for their formation. सबसे important जो character है carotenoids का, that is in carotenoids there is no requirement for the light for their formation. Light की इसमें कोई जरूरत नहीं होती है उनकी formation के लिए. Absorb blue and violet light. ये specially two lights को absorb करता है carotenoids होते हैं, और वो two lights जो these are blue and violet lights. Commonly they are known as the tetraterpenes, और commonly carotenoids को जो other words में जो है को क्या कहा जाता है? tetraterpenes. Carotenoids are of two types. Further, carotenoids are categorized into two categories, that are carotenes and the xanthophylls. That means carotenoids are further of two types, and these two types are the carotenes and the xanthophylls. Carotenes, they are reduced molecules. जो carotenes होते हैं, these are generally known as the reduced molecules, have the general formula, और इसमें जो general formula है carotenes का, that is C40H56. This is the general formula of the carotenes. Carotenes are of further three types. अब उसके बाद हम carotenes को further three categories में divide करेंगे, and these three types are the alpha, beta and gamma. That means carotenes are categorized into three categories. These are alpha, beta and gamma. First we will take the alpha carotenes. Alpha carotene is present in all the plants. Alpha carotene जो है that is present in all the plants, whereas beta carotene is the abundant precursor of vitamin A. जो gamma beta carotene है, it is most abundant precursor of the vitamin A. Gamma carotene in green sulfur bacteria. जो gamma carotene है, it is most abundant in the green sulfur bacteria. Next is the lycopene, that is red pigment in tomato and chili. अब जो lycopene है, it is red pigment and it is totally present in the tomato and chilies. So on the behalf of that reason, tomato as well as chilies, they are red in color due to the presence of the pigment, and the name of that pigment is the lycopene. Xanthophylls, yellow brown pigment, ये yellow brown pigment होते हैं and are oxygen containing derivative of the carotenes, और इसमें oxygen derivative होता है कैरोटीन का, जो कि ये जो है ज़ैंथोफिल जो है उसको कंटेन करते हैं. नेक्स्ट इज अबंडेंट इन वॉयला, और सबसे ज्यादा ये किसमें पाया जाता है? वॉयला में. नेक्स्ट इज फ्यूकोजेंथिन इन ब्राउन एल्गा. फ्यूकोजेंथिन जो है दैट इज प्रेजेंट इन द ब्राउन एल्गा. फाइकोबियंस, these are photosynthetic pigments of the some algal group. अब ये जो फाइकोबियंस हैं, जो नेक्स्ट टाइप ऑफ photosynthetic pigments की जो कि फाइकोबियंस है, and these are the photosynthetic pigments of some algal groups structurally related to the biopigments, और structurally किसके साथ relate करते हैं? ये biopigments के साथ. and there are open tetrapyrrole with neither magnesium nor phytol, और ये जो होते हैं ये open tetrapyrrole होते हैं and there are open tetrapyrrole, ये open tetrapyrrole होते हैं with neither magnesium, ना तो इनमें magnesium होता है ना ही phytol होता है. Only water soluble pigment, और ये सिर्फ water में ही soluble होते हैं, that means these are water soluble pigments. Next types, phycoerythrin. Phycobians are further of different types, that is phycoerythrin, phycocyanin and phycyclin. तो ये 3 टाइप्स के हमारे पास phycobians है. सबसे पहले हम देखा phycoerythrin, phycocyanin and allophycocyanin. So these are the 3 types of the pigments that are present in the phycobians. These are important accessory pigments in the blue, green alga and red alga. ये सबसे important accessory pigments हैं किस चीज के? Blue, green alga as well as red alga के. They are also called as billy proteins. इनको billy proteins भी कहा जाता है. Next is the absorption spectrum. What do you mean by the term absorption spectrum? The chloroplast pigments are responsible for the absorption of light. अब ये जो chloroplast pigments हैं, these are totally responsible for the absorption of light. A particular pigment absorbs light ray, और एक particular pigment जो होगा वो absorb करेगा light rays को, जिसकी की different wavelengths होंगी. For example, chlorophyll A absorb blue and red light. उदाहरण के तौर पर अगर हम देखें तो जो chlorophyll A है, it directly absorbs the blue and the red light in the visible spectrum. The amount of the light absorbed by pigment can be plotted. अब जो amount जो होगी light की, that can be absorbed, that is by the pigment, that can be plotted in the graph. उसको ग्राफ में प्लॉट किया जा सकता है. The graphic representation of the absorption spectrum. अब इसमें जो हमें graphic representation लेंगे absorption spectrum की against the wavelength, that is called as the absorption spectrum. अब ये जो graphic representation है absorption spectrum की against the, किसके against है? Against the wavelength, that is called as the absorption spectrum. So this is the graph, and this graph is plotted wavelength of the light, that is denoted in nanometer, and wavelength of light versus amount of the light absorbed. That means this graph is plotted between the wavelength of light versus amount of the light absorbed. So this is the wavelength of the light, that is 400 nanometer, 500, 600 and 700 nanometer, and this is the amount of the light absorbed against the wavelength. So this is the chlorophyll A, and yellow color, that light green color represents the chlorophyll B, and this yellow color that represents the carotenoids. So this is the graphic representation of the amount of the light that is represented against the wavelength of light as well as absorption amount of the light absorbed. So this absorption spectra of extracted chlorophyll and carotenoids and the primary light harvesting chlorophylls absorb light in the blue and the red region. इसमें जो light होगी that is absorbed in the blue and the red region, and carotenoids होंगे that absorbs in the blue and green region, और जो carotenoids होंगे that absorbs the light in the blue and the green regions. Next is the action spectrum. Photosynthesis takes place in the visible light. अब इसमें जो photosynthesis का जो process है, it takes place in the visible light, ranging from 400 nanometer to 700 nanometer, और उनकी जो wavelength है, जो light है, that ranges from 400 nanometer to 700 nanometer wavelength. It does not take place at the same rate. अब इसमें जो है ये same rate पर take place नहीं करता है. with other wavelengths. That means इनकी जो wavelengths होंगी, different wavelengths पर वो different place पर जो होगा वो occur करेगा. The rate of photosynthesis at different wavelengths. अब जो rate of photosynthesis का at different wavelengths पर is measured and plotted. उसको measure करेंगे as well as plotted करेंगे by the amount of the action. That is oxygen evolution, carbon dioxide utilization. और इसमें amount of action का जो मतलब है that is the oxygen evolution as well as carbon dioxide utilization. This graphic representation of rate of photosynthesis against wavelength is called as action spectrum. अभी graphic representation जो बन रही है rate of photosynthesis against the wavelength, that is called as the action spectrum. So this is the graph that is plotted between the action spectrum versus rate of the photosynthesis. तो ये एक्शन स्पेक्ट्रम का दिस ग्राफ शोज द रिप्रेजेंटेशन ऑफ द एक्शन स्पेक्ट्रम एंड दिस ग्राफ इज प्लॉटेड बिटवीन द वेवलेंथ वर्सेस rate of the photosynthesis. So this is the most active wavelength for the photosynthesis. जो peak point पर है, जो peak point पर जो हमारी wavelength बन रही है, that is the most active wavelength है for the process of the photosynthesis. And जो mid में है that is the process of the photosynthesis. And अगर हम down में जाए तो that is the absorption of light by the chlorophyll A. This absorption of light is absorbed by the chlorophyll A. So this is the graphic representation of the action spectrum, and this graph is plotted between wavelength as well as amount of the light, rate of the photosynthesis. Next, what is light reaction? What do you mean by the term light reaction? In 1882, T.W. Engelmann plotted the first action spectrum of photosynthesis using green alga, that is cladophora. तो सबसे पहले जो हमारे 1882 में T.W. Engelmann जो साइंटिस्ट थे, उन्होंने जो ग्राफ प्लॉट किया था फर्स्ट एक्शन स्पेक्ट्रम का ऑफ द फोटोसिंथेसिस, वो कैसे उन्होंने प्लॉट किया था यूजिंग द ग्रीन alga, एंड दैट ग्रीन alga इज द cladophora. The maximum rate of the photosynthesis occurs with the red light of the visible spectrum. अब जो maximum rate जो था photosynthesis का वो occur किया था with the help of the red light of the visible spectrum. Most of the photosynthesis takes place in the blue and the red region. Maximum photosynthesis का जो process था that takes place in the blue and the red region of the visible spectrum. Photosynthesis does not take place at the same rate. Photosynthesis का process जो है that does not take place at the same rate at the other wavelengths of the spectrum. This happens because, अब ऐसा क्यों होता है? Because the accessory pigments have only the capacity to absorb light energy. क्योंकि जो accessory pigments हैं, they have only the capacity to absorb the light energy and transfer it to the chlorophyll A. क्योंकि उन्हीं में एक ऐसी capability है या capacity है जो कि absorb करते हैं light energy को, and after that they transfer that energy to the chlorophyll A. The light reaction takes place only in the chlorophyll A. जो light reaction है, it takes place only in the chlorophyll A, that is the reaction center. Next is mechanism of photosynthesis. What mechanism that to be adopted for the process of the photosynthesis? Photosynthesis is an oxidation reduction reaction. Photosynthesis एकमात्र एक ऐसी process है जिसमें oxidation reduction जो होती है वो reactions involve होती हैं in which water is oxidized, जिसमें कि water जो होता है वो oxidized होता है and carbon dioxide is reduced. तो हमने कहा कि photosynthesis is the only process in which both oxidation and reduction processes takes place and in which water is oxidized and carbon dioxide is reduced. The general equation that represents photosynthesis may be written as, अब general equation जो है that represents the process of the photosynthesis may be written in the following manner. That is 6 molecules of carbon dioxide plus 12 molecules of water. They give rise to C6H12O6, that is glucose, plus 6 molecules of water and plus 6 molecules of the oxygen. So this is the general representation of the process that represents the process of the photosynthesis. The end product of photosynthesis are carbohydrate, water and oxygen. अब जो end product बना है किस चीज का? फोटोसिंथेसिस का. That is carbohydrates जो कि ग्लूकोज की फॉर्म में है plus water and oxygen. So this is the end product of the process of the photosynthesis. Carbon dioxide and water are the raw materials for the photosynthesis. अब जो कार्बन डाइऑक्साइड है as well as water जो है these are the raw materials for the process of the photosynthesis. So the raw materials for the process of the photosynthesis are carbon dioxide and water. So these are carbon dioxide and water. So these are the raw materials for the process of the photosynthesis. Oxygen is liberated into the atmosphere as a byproduct during this process. जो ऑक्सीजन जो है वो लिबरेटेड होगा कहां पर, एटमॉस्फेयर में लिबरेटेड होगा एज अ बाय प्रोडक्ट ड्यूरिंग द प्रोसेस. दिस इवॉल्व्ड ऑक्सीजन कम्स फ्रॉम वाटर. अभी जो इवॉल्व्ड ऑक्सीजन है दैट इवॉल्व्ड ऑक्सीजन कम्स फ्रॉम द वाटर एंड नॉट फ्रॉम द कार्बन डाइऑक्साइड. अभी ये जो इवॉल्व्ड ऑक्सीजन है it directly comes from the water, not from the carbon dioxide. In photosynthetic plants, the hydrogen donor is water. अब photosynthetic plants में जो donor है, जो hydrogen donor है, that is the water. The ultimate source for the hydrogen donor in the process of the photosynthesis is water, and the source of oxygen released is also as water. अब जो oxygen का जो source है, the source of oxygen, it is also released in the form of water. जो oxygen release source होगा वो भी क्या है, water. In 1932, रॉबर्ट एमर्सन एंड अर्नोल्ड. अब 1932 में दो साइंटिस्ट थे, रॉबर्ट एमर्सन एज वेल एज अर्नोल्ड. उन्होंने क्या प्रपोज किया था, दैट द होल प्रोसेस ऑफ द फोटोसिंथेसिस, जो होल प्रोसेस है फोटोसिंथेसिस का, इन्वॉल्व्स टू डिस्टिंक्ट फेजेस. अब ये दो साइंटिस्ट जो थे, इन्होंने क्या जो था कंक्लूजन निकाला था, उन्होंने जो कंक्लूजन निकाला था कि जो होल प्रोसेस है फोटोसिंथेसिस का, दैट टेक्स प्लेस इन द टू फेजेस. दो फेजेस में टेक्स प्लेस करेगा, देयर फोटोकेमिकल फेज एज वेल एज बायोसिंथेटिक फेज. और जो दो फेज थे वो थे फोटोकेमिकल फेज एज वेल एज बायोसिंथेटिक फेज. पहले हम लेते हैं फोटोकेमिकल फेज और द लाइट रिएक्शन. It is the light dependent phase of the photosynthesis. ये light dependent, so that means that this phase is totally depends upon the light. So this is the light dependent phase of the photosynthesis in which ATP and NADPH are formed in the presence of light. इसमें दो products बनेंगे, that are the ATP as well as NADPH in the presence of sunlight. It takes place in the grana of the chloroplast. और especially कहाँ takes place करेंगे, in the grana of the chloroplast. It takes place in the presence of sunlight. और ये sunlight की presence में takes place करेंगे. It consists of the following step. अभी जो light reaction है या photochemical phase है, it takes place the following steps. अब कौन कौन से following steps है? First step is the light absorption, second is the water splitting, and the third is the oxygen release, and fourth is the formation of high energy chemical intermediates. इसमें जो formation होगी that is the high energy chemical intermediates, और ये जो high energy chemical intermediates हैं that are in the form of ATP and NADPH. In 1935, Robert Emerson proved that two pigment systems are involved in the light reaction. तो 1932 में जो एमर्सन रॉबर्ट एमर्सन थे, उन्होंने प्रूव कर दिया कि ये जो दो पिगमेंट्स हैं, these are involved in the process of the light reaction. So the reaction center, they are pigment system 1, that is photosystem 1, and pigment system 2, that is PS2. तो दो उन्होंने जो सिस्टम कहा कि that are necessary for the process of the light reaction, वो कौन कौन से थे? एक था pigment system 1 और दूसरा जो था pigment system 2, या PS1 and PS2. The reaction center and the light pigment system 2. The reaction center and the light harvesting system together forms photosystem or the pigment. अब ये जो reaction center है, ये light harvesting system जो है, जो light harvesting system as well as reaction system दोनों मिलकर क्या बनाते हैं? accessory pigments. ये together दोनों मिलकर क्या बनाते हैं? photosystem या pigments को बनाते हैं, pigment system को. About 250 to 450 pigment molecules constitute a single photosystem. तो नियर अबाउट एक फोटोसिस्टम के लिए कितने पिगमेंट मॉलिक्यूल्स का होना जरूरी है? नियर अबाउट 250 से लेकर 450 पिगमेंट मॉलिक्यूल्स का होना जरूरी है एक सिंगल फोटोसिस्टम की फॉर्मेशन के लिए. फोटोसिस्टम्स आर फंक्शनल एंड स्ट्रक्चरल यूनिट्स. अभी जो फोटोसिस्टम्स हैं, दीज आर द स्ट्रक्चरल एंड फंक्शनल यूनिट्स कंसिस्टिंग ऑफ प्रोटीन कॉम्प्लेक्सेस, जो कि कंसिस्ट करते हैं प्रोटीन कॉम्प्लेक्सेस को, involved in the photosynthesis, जो कि कौन से प्रोसेस में इन्वॉल्व होते हैं? They directly involved in the process of the photosynthesis. They are located in the thallopoids membranes of the plants. ये कहाँ locate करते हैं? They directly locate in the thallopoids membranes of the plants and alga or in the cytoplasmic membrane of the photosynthetic bacteria. टू काइंड ऑफ फोटो सिस्टम्स आर प्रेजेंट। अब दो काइंड्स के फोटो सिस्टम्स प्रेजेंट हैं। अब दो काइंड के फोटो सिस्टम्स कौन कौन से हैं? फोटो सिस्टम 1 एज वेल एज फोटो सिस्टम 2। दीज टू टाइप्स ऑफ द सिस्टम्स आर इन्वॉल्वड इन द फोटोसिंथेटिक प्रोसेस। बोथ फोटो सिस्टम्स आर रिक्वायर्ड फॉर द ऑक्सीजेनिक फोटोसिंथेसिस। अब ये दोनों टाइप्स के फोटो सिस्टम जो हैं दे आर डायरेक्टली इन्वॉल्वड फॉर द प्रोसेस फॉर द दैट इज ऑक्सीजेनिक फोटोसिंथेसिस। The photosystem 2 is located in the appressed region of the granule thylakoids। अब ये जो photosystem 2 है ये कहाँ लोकेट करता है? appressed region of the granule thylakoids। photosystem 1 is found in the non-appressed region। अब ये जो photosystem 1 है it is found in the non-appressed region of the grana and stroma thylakoids। अब कौन कौन से सिस्टम्स इसमें जो मेनली इन्वॉल्व है दैट इज द इलेक्ट्रॉन ट्रांसपोर्ट। सबसे पहले हम जो इसमें अब डिस्कस करेंगे द मोस्ट इंपॉर्टेंट टॉपिक ऑफ दिस चैप्टर दैट इज फोटोसिंथेसिस इन हायर प्लांट्स इज द इलेक्ट्रॉन ट्रांसपोर्ट सिस्टम। नॉन साइक्लिक फोटोफॉस्फोराइजेशन इट इज आल्सो टर्म्ड एज जेड स्कीम। अभी जो नॉन साइक्लिक फोटोफॉस्फोराइजेशन है इसको जेड स्कीम भी कहा जाता है। बोथ द सिस्टम्स आर इन्वॉल्व दैट इज photosystem 1 and photosystem 2 are involved in non-cyclic photophosphorylation। photosystem 2 consists of chlorophyll A660। अब जो photosystem 2 है वो क्या कंसिस्ट करता है? That consists the chlorophyll A660, chlorophyll A673, chlorophyll A690, chlorophyll B, chlorophyll C और chlorophyll D, carotenoids and phycobilins। क्लोरोफिल A680 is the reaction center। अब क्लोरोफिल A680 जो है that is the reaction center। It occurs in the stroma lamella। अब ये कहाँ करता है? stroma lamella में। The electron do not go back to the reaction center। अब जो इलेक्ट्रॉन है they do not go back to the reaction center but rather are accepted by the NADP positive। Photolysis of water uses up the electrons। अब ये जो photolysis of water है it directly uses up the electrons and leads to the formation of ATP as well as NADPH2। The steps in the process begins with the photosystem 2। अब कौन से ऐसे steps हैं जो photosystem 2 की process में involved होते हैं? Electrons are passed to plastoquinone producing substance। अब ये जो electrons हैं they directly passed into the plastoquinone producing substance that is the PQRS। PQRS that is the plastoquinone producing substance passes them to plastoquinone जो कि plastoquinone में पास होते हैं which passes them to the cytochrome system। उसके बाद वो कहाँ पास होते हैं? They directly pass into the cytochrome system। The cytochrome system passes them to plastocyanin which in turn passes them to the photosystem 1। अभी cytochrome system जो होगा वो दोबारा कहाँ पास करेगा? प्लास्टोसाइनिन में पास करेगा। उसके बाद वो कहाँ पास करेगा? After that they pass into the photosystem 1। The step afterwards includes FRS, ferredoxin and NADP reductase। अब उसके बाद जो steps afterward include होंगे that are the FRS, ferredoxin as well as NADP reductase। Next is photolysis of water। What do you mean by the process of photolysis of water? P680 becomes a strong oxidizing agent। अब जो P680 है it becomes the strong oxidizing agent। क्यों strong oxidizing agent है? And it splits the molecules of water। उसके बाद ये water molecules को split करता है to release the oxygen। जब water molecule वहां से split हो जाता है तो split होने के बाद वो क्या release करता है? It directly releases the oxygen। This light dependent splitting of water molecules is called photolysis of water। अब ये जो light dependent splitting of water molecule है it is directly known as the photolysis of water। Manganese, calcium and chloride ions plays an important role in the photolysis of water। अब कौन से important ions हैं जो photolysis की process में important role play करते हैं? That are the manganese, calcium and chloride। All these ions they have to play an important role in the process of the photolysis of water। The photolysis of water in photosynthesis was first demonstrated by Robert Hill in 1937। सबसे पहले जो photolysis of water था in the process of photosynthesis that was demonstrated by the scientist and this scientist was known as Robert Hill in 1937। Water is splitting into hydrogen ions। उसके बाद water जो है it directly splits into the hydrogen ions, oxygen as well as electrons। ऑक्सीजन इज वन ऑफ द एंड प्रोडक्ट ऑफ द फोटोसिंथेसिस, और जो एंड प्रोडक्ट है फोटोसिंथेसिस का दैट इज द ऑक्सीजन. सेकंड इज द साइक्लिक फोटोफॉस्फोराइजेशन एंड नॉन साइक्लिक फोटोफॉस्फोराइजेशन, also known as light reaction, Hill reaction और photochemical reaction. ज्यादातर इसको जो है और क्या नाम दिए जाते हैं, that is the light reaction, Hill reaction, photochemical reaction, generation of the assimilatory powers as well as photophase. In cyclic photophosphorylation only photosystem 1 works. तो cyclic photophosphorylation में अगर देखा जाए तो सिर्फ और सिर्फ फोटोसिस्टम 1 जो है वो वर्क करता है. फोटोसिस्टम 1 कंसिस्ट ऑफ क्लोरोफिल A670. अब फोटोसिस्टम 1 जो है क्या कंसिस्ट करता है, क्लोरोफिल A670, क्लोरोफिल A683, क्लोरोफिल A695, कैरोटीनॉइड्स, some molecules of chlorophyll B and reaction center that is 700 or P700. Next, this forms of photophosphorylation occurs on the thalloid membrane. इस टाइप का जो photophosphorylation का जो प्रोसेस है, it occurs only in the thalloid membranes. The electron begins photosystem 1. अब इलेक्ट्रॉन जो वो कहाँ begin करेंगे, photosystem 1 में, passes from the primary acceptor to ferredoxin, और कहाँ फिर पास करेंगे, primary acceptor to ferredoxin reducing substance, that is FRS, that is ferredoxin reducing substance. Then to ferredoxin, उसके बाद ferredoxin से then to cytochrome B6F and then to plastocyanin before returning to photosystem 1. This process produces a proton motive force. अब ये जो प्रोसेस है ये क्या प्रोड्यूस करता है, proton motive force. proton motive force को ये प्रोड्यूस करता है pumping H positive ions, और H positive ions को पंप करता है across the membrane, thereby generating concentration gradients that can be used to power ATP synthesis during the chemoosmosis. तो उस दौरान जो है ये ATP synthesis during the process of chemoosmosis के दौरान ये प्रोड्यूस करता है. Cyclic photophosphorylation neither produces oxygen nor NADP. अब जो cyclic photophosphorylation का प्रोसेस जो है it neither produces oxygen as well as NADPH. NADP positive does not accept the electrons. NADP positive जो है it does not accept the electrons. They are instead sent back to cytochrome. उसकी जगह पर वो उसको दोबारा कहाँ बैक करता है, साइटोक्रोम B6F कॉम्प्लेक्स में, B6F कॉम्प्लेक्स में उनको दोबारा पास आउट करता है. This process is mostly seen in the bacteria, and this process is directly seen in the process of bacteria and favored in anaerobic condition. Next is chemoosmotic hypothesis. What do you mean by the process chemoosmotic hypothesis? ATP synthesis in the chloroplast is explained by chemoosmotic hypothesis. अब जो ATP synthesis है chloroplast में, that is explained with the help of chemoosmotic hypothesis. It was proposed by Peter Mitchell in 1961, और इसको किसने प्रपोज किया था, Peter Mitchell in 1961. According to chemoosmotic hypothesis, इस hypothesis के according during photosynthesis a proton gradient, जब photosynthesis का process होगा उधर proton gradient होगा that is developed across the thalloid membrane. Protons are accumulated towards the inside of the thalloid membranes in the thalloid lumen. अब जो protons जो है they are accumulated towards the inside of the thalloid membranes in the thalloid lumen. The proton gradient is caused by 3 waves. अब जो proton gradients हैं these are caused by the 3 steps, and these 3 steps are water molecule is broken up to H positive and OH negative ions. अब जो water molecules हैं they are directly broken into H positive as well as OH negative ions on the inner side of the thalloid membranes. As a result, H positive are accumulated within the thalloid lumen. अब जो H positive होंगे they are directly accumulated with the thalloid lumen. When the electron moves, दो photosystem 1 and photosystem 2, जब इलेक्ट्रॉन जो होगा वो मूव करेगा with the help of photosystem 1 and photosystem 2, H positive are transported across the thalloid membrane. तो जो H positive ions होंगे they are directly transported across the thalloid membranes. Primary electron acceptor is located towards the outer side of the thalloid membranes. अब जो primary electron acceptors हैं they are located towards the outer side of the thalloid membranes. दिस प्राइमरी इलेक्ट्रॉन एक्सेप्टर ट्रांसफर, अभी जो प्राइमरी इलेक्ट्रॉन एक्सेप्टर है इट ट्रांसफर्स द इलेक्ट्रॉन टू ए H कैरियर एंड नॉट टू बी एन इलेक्ट्रॉन कैरियर डायरेक्टली। H कैरियर होगा जबकि नॉट एन इलेक्ट्रॉन कैरियर डायरेक्टली। This hydrogen carrier molecule transfers the electron to the electron carrier. अब ये जो hydrogen carrier molecule है, it transfers the electron to the electron carrier, that is cytochrome B, cytochrome F, etc. The inner side of the thylakoid membranes and removes H positive ions from the stroma. अब जो inner side होगी thylakoid membranes की, वो remove करेगी H positive ions को from the stroma while transporting an electron. This proton is released into the inner side of the thylakoid membrane. अब ये जो proton होगा वो released करेगा into the inner side of the thylakoid membranes. Third number is NADP reductase enzyme is located on the stroma. अब जो NADP reductase enzyme है, it is located on the stroma side of the thylakoid membranes. When electrons come from the electron acceptor, अब ये जो electrons हैं जब वो कहाँ से आएंगे, comes from the electron acceptor of photosystem 1, H positive are necessary for the reduction of NADP positive. अब जो H positive है, it is very necessary for the reduction of NADP positive to NADP H plus H positive, and these H positive are removed from the stroma. अब ये H positive ions होंगे, they directly removed from the stroma. By these ways within the chloroplast, इन ways से जो कि ये ways जो within the chloroplast present हैं, H positive are accumulated in the thylakoid lumen. जो H positive होंगे, they directly accumulated in the thylakoid lumen and decreased in number in the stroma. और इनका जो number होगा stroma में वो क्या कर जाएगा, decrease कर जाएगा. This creates a proton gradient across the thylakoid membrane. अब ये जो है वो क्या क्रिएट करेंगे, प्रोटोन ग्रेडियंट एक्युमुलेट करेंगे अक्रॉस द thylakoid membrane. As a result, pH is decreased in the thylakoid lumen. और इसके कारण जो रिजल्ट होगा, that is pH is decreased in the thylakoid lumen. The proton gradient is broken when the proton moves to the stroma. अब वो जो proton gradient है, it directly brokens और ये कब broken करेगा, when the proton moves to the stroma. जब जो proton होगा वो move करेगा towards the stroma through the transmembrane channel of the F0 of the ATPase enzyme across the thylakoid membrane. So this is the ATP synthesis through the process of chemoosmosis. This is photosystem 1. This is cytochrome B and F, and here there is the release of the H positive ions, and this is the photosystem 2, and these H positive ions are released from the cytochrome B and F, that is these are the H positive ions. And in photosystem 1 there is the energy, that is the NADP positive and NADPH. This is the lumen, and this is the ATP synthesis, and this is the F1, and this is the energy carrier, that is AGP is directly transformed to the ATP, adenosine diphosphate to adenosine triphosphate. And this is the thylakoid membrane. This pink colored membrane is known as the thylakoid membrane, and these are the stroma that are present in the thylakoid membrane. Next is ATPase enzyme has two parts. अब जो ATPase enzyme है इसके दो parts हैं. The base of the ATPase is called F0 and head piece is called F1. अब जो base है ATPase का उसको हम F0 बोलेंगे और जिसका head piece है that is called as the F1. F0 forms a transmembrane channel. अब ये F0 जो है it directly forms the transmembrane channel which is embedded in the thylakoid membrane on the side that faces the stroma. Chemoosmosis requires a membrane. Chemoosmosis जो होगा it directly requires a membrane, a proton pump, a proton gradient, and ATPase enzyme. The channel of the F0 of the ATPase allows the diffusion of H positive back. अब ये जो channel है F0 of the ATPase ये allow करेगा diffusion of H positive ions को back across the thylakoid membranes. As a result, enough energy is released. और उस कारण जो होगा वो enough energy जो होगी या ज्यादा से ज्यादा energy जो होगी वो वहां से released होगी. This energy activates ATPase और वही energy जो ज्यादा से ज्यादा energy released होगी वही energy जो होगी उस ATPase को क्या करेगी, activate करेगी. ATPase catalyzed ATP synthesis from ATP. अब ये ATPase जो होगा वो catalyze करेगा ATP synthesis from ADP. This ATP is used immediately in the dark reaction. अब ये ATP जो है it directly used in the dark reaction that taking place in the stroma, जो कि कहाँ take place करता है, stroma में. As a result, sugar synthesis takes place by carbon dioxide fixation. और इस कारण जो होगा बहुत sugar synthesis जो होगा that takes place during the carbon dioxide fixation. So this is the diagram, and this diagram shows the outer side of the membrane. Outer side of the membrane, and this is the ATP molecule, and this ATP molecule channelized into two parts, that is the ADP and pi, and this is the matrix, and this is the inner membrane part, and this is the 2H positive ions, and below this is the inner mitochondrial membrane. So this is all about this part number 2, and our next topics are to be continued in our next lecture, that is part number 3.